Welcome to lesson 1.4 in Unit 1 Cell Biology. And in this lesson, we're going to be covering membrane transport and going over how certain substances and compounds in actual cell membranes are transported across the bilayer. So let's get into it. So, as we know from the previous lesson, the phospholipid bilayer is selectively permeable. And what this essentially means is that some molecules can pass through the bilayer easily, others need energy to get through. And the movement of materials across a membrane may occur either actively or passively. All right, so passive transport. So passive transport involves the movement of material or substance along a concentration gradient. So it's the key word, it's along a concentration gradient. And this is usually from a high concentration to a low concentration, and it requires no energy during the process or ATP. And there are three types of passive transport. There's simple diffusion, which is the movement of small molecules across a membrane, osmosis, which is the movement of water molecules across a membrane, and facilitated diffusion, which is the movement of large or charged molecules via membrane proteins in the phospholipid bilayer. All right, so simple diffusion. So simple diffusion is the passive net movement of particles from an area of high concentration to an area of low concentration. And simple diffusion occurs down the concentration gradient until both concentrations are at an equilibrium. And simple diffusion does not require any form of energy or ATP in order to function or occur. All right, osmosis. Osmosis is the net movement of water from a low solute to a high solute concentration. And just some key terms: a solute is a substance that dissolves. Uh, a solute is a substance that is dissolved in a solvent, and a solvent is a liquid that dissolves a solute. And water is known as the universal solvent. So as we can see here in this diagram, in a liquid that is low in sugar concentration or low in solute concentration and high in solvent concentration, this is going to move to a liquid or a solution that is high in solute concentration through a selectively permeable membrane. All right, osmolarity. So osmolarity is the measure of solute concentration in a solution. Solutions can be categorized with three criteria. They can be categorized as hypertonic, hypotonic, or isotonic. Hypertonic solutions are solutions with a high solute concentration. Hypotonic solutions are solutions with a low solute concentration. And isotonic solutions are solutions that have the same solute concentration. So moving on to the importance of osmotic control. So in a solution with higher osmolarity, water will rapidly leave the cell and their cytoplasm will shrink, leaving the cell in a shriveled state, or a plasmolyzed state if it's a plant cell. However, in an isotonic solution, water molecules will enter and leave the cell at the same rate, and the cell will remain healthy and in a normal state if it's an animal cell, or a flaccid state if it's a plant cell. In hypotonic solutions, or solutions with lower osmolarity, the cell will take in water rapidly and will swell, and it may burst or lyse as a result of this. In animal cells, this will be known as being in a lyse state, and in plant cells, this will be known as being in a turgid state. Although do keep in mind, plant cells actually cannot burst because of their cell wall, so do keep that in mind for plant cells. All right, moving on to facilitated diffusion. So this is the passive movement of molecules across membranes via membrane proteins. And there are two types of membrane proteins that are used primarily in facilitated diffusion. You have carrier proteins, which are slower in their actual channeling of the substance or compound. And then you have channel proteins, which are faster in their transportation and movement of the compound. The diameter and chemical properties ensure only one type of particle can pass through a membrane protein. So, for example, sodium may be able to pass through, or potassium may be able to, but not both simultaneously. So, moving on to active transport. So, active transport uses energy, or ATP, to move molecules and substances against, so keyword being against, a concentration gradient. And during active transport, globular proteins will often be used that are often called pump proteins. And the iron or molecule can pass to the opposite side of the membrane through these pump proteins. All right, so as an example, um, being sodium and potassium pumps in axons. So axons are part of neurons, and they convey messages rapidly in an electrical form called a nerve impulse. The sodium potassium pump will follow a repeating cycle of steps that results in three sodium ions being pumped out of the axon and two potassium ions being pumped in. And so this pump is an example of both active and passive transport. And we can see this in the image here. So we can see there are three sodium ions being pumped out of the actual axon in the neuron, and then two potassium ions will be pumped inside of the neuron. So facilitated diffusion of potassium in axons. So potassium channels in axons are gated, sorry. And if an axon has more positive charges outside than inside, the potassium channels will be closed. And at one stage during the nerve impulse, there are going to be more positive charges inside of the axon than outside, causing the potassium channels to open and the potassium to diffuse out. You can see this here in this image right here in the sodium pump. All right, so active transport in sodium and potassium pumps. So three sodium ions will bind to the pump, and a phosphate group will be transferred to the pump via the breaking down of ATP. Because of this, the pump will undergo conformational change. This will lead to sodium being pumped across the membrane, and the pump will stay open to the on one side until two potassium ions will bind, and then those potassium ions will be diffused across the membrane inside of the cell. You can see that here in this image as well. So you're going to have three sodium ions binding into the actual pump. It's going to go. The pump will undergo conformational change due to a phosphate group. The sodium ions will be pumped across the cell membrane to outside of the cell, and they'll be expelled. And then two potassium ions will bind into the actual pump, and then they're going to be diffused into the actual cell across the membrane. All right, vesicular transport. So materials destined for secretion or excretion out of the cell are transported around the cell in vesicles. And there are two types of vesicular transport in cells. You have exocytosis, which is the transporting of materials outside of the cell. Notice exo, external, so outside. And then endocytosis, which is the transporting of materials inside of the cell. Endocytosis. So in endocytosis, a vesicle is formed on the inside of the plasma membrane, and thus this will allow material to be taken into the cell. The material can contain water and solutes from outside of the cell. They also often contain larger molecules that are needed by the cell that cannot pass through the plasma membrane. This process, so how this process occurs is that a small region of the membrane is pulled from the rest of the membrane and is pinched off. And then proteins use energy or ATP to carry out this process. All right, exocytosis. So this is when a vesicle fuses with the plasma membrane and releases its contents outside of the cell. The vesicles can be used to release materials from the cell after proteins have made it in the rough endoplasmic reticulum and processed it in the Golgi, and they can be released. Or exocytosis can also occur to expel waste products and also other unwanted materials. And here's just an image that just exemplifies and sort of consolidates the process of endo and exocytosis. So that's going to be it for lesson 1.4, membrane transport. Thank you for listening, and I'll see you next time.